Hi, and thank you for having me. It's a real pleasure to have the opportunity to talk about one of my PhD projects here. I'll start with a bit of an introduction of why we should care about extrachromosomal DNA in cancer. So extrachromosomal DNA was first described in the 1960s, where they said we have encountered a new anomaly, the presence of very small double chromatin bodies, sometimes in large numbers. And you can see here a karyotype, and what back then was called double chromatin bodies and then double minutes is what we now call extrachromosomal DNA, or short ecDNA. So ecDNA essentially are just these freely floating circles in the nucleus next to the chromosomes. They originate from chromosomes, and in cancer, where we get lots of DNA damage events, like, for example, 2 DNA double-strand breaks or even more severe chromothripsis, a bit of the chromosome is excised and religates onto the circle, so you end up with ecDNA and a shortened chromosome. For the case in this project, the ecDNA originates from chromosome 8, and particularly it harbors the c-MYC oncogene. Just for reference, it's around 1.5 megabases in size. Now, when we look at cell division, ecDNA presents a special case. They don't have centromeres, so during cell division, they can't be attached to the spindle apparatus, and we can end up with random segregation of the ecDNAs, which then means we'll get a lot of heterogeneity, and also we can accumulate lots of amplifications of an oncogene, which means that ecDNA also drives rapid tumor evolution. And recently it's been discovered that ecDNA is actually associated with cancer. So 17% of all newly diagnosed cancers have ecDNA in the cancer cells. And some cancers like glioblastoma, even 60% of all cancer cases have ecDNA. So to study the ecDNA, we use a cell line which was established in the 1970s. And they cultured the tumor tissue and found that the cell line, which was just numbered 320, contained these circles, or ecDNA shown in pink. And then one of the batches, over time, actually, the ecDNA disappeared. And what they instead found was this homologously staining region, short HSR. So here we have really the perfect model system to study ecDNA. And then from the same tumor background, the ecDNA reintegrated into chromosome context. So to study DNA replication, we use Oxford Nanopore Technology sequencing slightly differently. And we've heard a lot about detecting modifications. And we actually use DNA thymidine base analogues. So these are thymidine analogues that don't naturally occur. We culture the cells first in EdU, which is shown in blue, and then in the second thymidine base analog, which is called BrdU, shown in pink, and then the DNA is sequenced. And we have a software which we call DNAscent, and now we can not only detect the 4 natural bases and any naturally occurring modifications, but also in thymidine positions differentiate between EdU, BrdU, and thymidine. And just very high level, how this works is we feed the raw signal into our residual neural network, and then for each thymidine position, we get the probability of the thymidine either being a natural thymidine, a BrdU, or an EdU. And if we look at a sketch of DNA replication, we start with the DNA double-strand template on top. Then at positions that we call origins of replication, the DNA is opened up, and 2 replication forks progress into opposite directions. Now at the beginning, the cells are in the blue medium. So we'll incorporate the blue base analog, or EdU. Then they are transferred to pink, so we get a bit of pink afterwards. And we can end up with these traces. And as we know how long the cells were in each base analog, we can then infer how quickly the replication forks progressed. Now recently, we added a new feature to also automatically detect if a replication fork hits an obstacle and erroneously stops or stalls. And how you can imagine this is, I'm just showing one DNA strand to make it simple. If the replication fork stops, here the rightward moving fork stops, the concentration of the base analog in the cells lowers while the stall is resolved, and then we get this sudden drop-off in signal. And to show you some real data, on the top, the 4 tracks are one read, so we get the segmentation into EdU and BrdU in blue and pink, and then the raw signal. And the top fork is moving to the left, and you see in the left, in the pink one, you see a sudden drop-off in BrdU probability. So this means that this replication fork was stalled. Compared to the bottom, where you also see a leftward moving replication fork, but here you see a smooth run out of the signal. So this fork didn't hit any obstacles and was very unlikely stalled. We also perturb DNA replication with a drug that is called hydroxyurea, or short HU. And just very briefly, what this does is essentially it depletes the nucleotide pool by blocking the synthesis of DNTPs. And so with that, we have everything we need to study whether DNA replication dynamics differ on those extrachromosomal DNA circles. We use our DNAscent software and ONT sequencing with the Promethean sequencer. First, I was interested in looking at the DNA replication dynamics genome-wide. So here I'm looking at all chromosomes, comparing the cell line with ecDNA in pink to the one without in blue. And we can appreciate that overall the replication forks moved slower in the cell line that has the ecDNA. Looking at stalling, there was more replication fork stalling going on in the cell line with the extrachromosomal DNA circles. Now moving on to comparing in the same cell line the ecDNA to the rest of the chromosomes, we see that in ecDNA or on ecDNA, again, the fork progression rates are significantly slower compared to the chromosomes, while again, there was more replication fork stalling. So next, I'm looking at what happens if we treat the cells with hydroxyurea. So here I'm just showing the reference circle that we assembled for the ecDNA and a few genes for reference. You can see in orange the c-MYC locus. And first, we are looking at fork speed from slow in blue to pink in fast-moving replication forks. This is averaged in 20 kb segments. And if you just relax your eyes a bit and zoom out, you probably appreciate that there's more blue on the right-hand side under HU treatment. This is what we'd expect. And we can also look at the stall scores. This is a bit more tricky. The stall score goes from 0 unstalled to 1 likely stalled. And of course, we then wanted to quantify this. So we see that under HU treatment, as expected, the replication forks did slow down. And this is unexpected. We also saw less stalling. Previously, it was not possible to really differentiate between slower fork progression and stalling due to a lack of methods. But now we can really say that it's slower forks, but not more stalling caused by hydroxyurea. Then finally, I was interested in checking whether hydroxyurea had a stronger effect on the extrachromosomal DNA compared to the chromosomes. And for this, I use a bootstrapping method, and I'm comparing how severely is ecDNA affected compared to the chromosomes. And I do indeed find that with respect to the fork progression or fork speed, ecDNA was more severely impacted by HU treatment. For the stall scores, I did not find a significant difference. So with that, I'd like to wrap it up and say that with our AI-based software DNAscent, we were able to study for the first time DNA replication dynamics on ecDNA at very high resolution and throughput. And as ecDNA is a driver of tumor evolution and tumor heterogeneity, it's really useful to have these maps of ecDNA replication dynamics, including fork speed, stalling, and origin usage. I didn't show the origin usage today. And we really hope that our methodology paves the way towards targeting ecDNA as the vehicle of oncogene amplifications in cancer. So with that, I'd like to thank my supervisor, Mike, the collaborator, Chadwick, who did all the wet lab work, and his supervisor, Julian. And I'm looking forward to your questions. Thank you.